Welcome back to the channel. In this video, we're going to be following on from the previous video and discussing the remainder of the organelles found within eukaryotic cells. So first, let's explore the endoplasmic reticulum, a key organelle in eukaryotic cells that exists in two forms, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum is covered with ribosomes on its outer surface, giving it a rough appearance under a microscope. These ribosomes are essential for protein synthesis. The rough endoplasmic reticulum provides a large surface area for assembling proteins and glycoproteins, which are vital for cell structure and communication. Additionally, it acts as a transport network, moving these molecules through the cell. For example, proteins synthesized in the RER are often packaged into vesicles and sent to the Golgi apparatus for further processing. The SER, or smooth endoplasmic reticulum, lacks ribosomes, making it appear smooth and more tubular under a microscope. It specializes in the synthesis, storage, and transport of lipids and carbohydrates. These molecules play critical roles in energy storage, membrane formation, and signaling pathways. For instance, the SER in liver cells is involved in detoxifying harmful substances, while in muscle cells, it helps regulate calcium ion levels for muscle contraction. The Golgi apparatus is a vital organelle, often described as the cell's post office. Its primary role is to receive, sort, package and dispatch proteins and lipids produced by the endoplasmic reticulum. Here's how it works. Proteins and lipids made in the endoplasmic reticulum are transported to the Golgi apparatus in small vesicles. Once inside, they are processed and modified. For example, the Golgi can add carbohydrates to proteins, forming glycoproteins, which are important for cell signaling and structure. Similarly, it processes lipids to prepare them for their roles in the cell membrane or energy storage. After modification, these proteins and lipids are packaged into Golgi vesicles. These vesicles then move to their destination, which could be within the cell or the cell membrane. If they're meant to leave the cell, the vesicles fuse with the membrane and release their contents outside, a process called exocytosis. Beyond packaging and transport, the Golgi apparatus is also crucial for creating lysosomes, which are involved in breaking down waste and recycling cellular material. Okay, so there are four more organelles that are commonly found within eukaryotic cells. The first one we're going to discuss is lysosomes. These are small vesicles filled with powerful hydrolytic enzymes. They play a vital role in the immune system by digesting harmful materials like bacteria and debris ingested by white blood cells. Lysosomes can also release enzymes outside the cell through a process called exocytosis, breaking down materials in the cell's surroundings. Inside the cell, they act like a recycling center, breaking down worn-out organelles so their components can be reused. In some cases, lysosomes can completely digest dead cells, ensuring waste is removed efficiently. Next up, we have ribosomes, the protein factories of the cell. These tiny organelles are made up of two subunits and come in two forms, 70S ribosomes in prokaryotic cells and 80S ribosomes in eukaryotic cells. Ribosomes are the site of protein synthesis, where amino acids are assembled into proteins based on genetic instructions from mRNA. Moving on to the cell wall, a defining feature of plant and fungal cells. In plants, the cell wall is made up of cellulose microfibrils, which provide mechanical strength, helping the cell resist bursting under high water pressure during osmosis. Beyond individual cells, the cell wall supports the entire plant, giving it structure and rigidity. In fungi, the cell wall is made of chitin, a strong but flexible material that also contributes to structural support. Finally, let's discuss vacuoles in plant cells. These large, fluid-filled organelles serve multiple purposes. They help maintain turgor pressure, which keeps plant cells firm and provides structural support to leaves and stems. Vacuoles also act as storage centers, containing sugars and amino acids that the plant can use as a food source when needed. In summary, these organelles and structures work together to support the cell's functions. Lysosomes manage waste, ribosomes build proteins, cell walls provide strength, and vacuoles ensure plants stay upright and nourished. Each plays a specialized yet interconnected role in maintaining cellular health and overall function. This concludes part 2 on our mini-series on eukaryotic cells. Thank you very much for taking the time to watch. Make sure you like, comment and subscribe. Also, feel free to check out all the other videos on this channel. Thank you very much for watching.